some of the databases having some other tools which to analyze the data. So some databases will provide you the information, the data, and how to retrieve the data. You may not able to analyze the data using those databases. So some of the database they will provide, they will be having other tools to analyze the data too. Okay, and they will be linked to that also. And yeah, today we will see one of the the tool analysis of the sequence information, that is pairwise sequence alignment. Okay, and yeah, today's lecture outline is why we need to compare the sequences and what is the pairwise sequence analysis. And thus pairwise sequence analysis, one of it is dot plot. What is that? And then dynamic programming algorithms, which is more useful and that is frequently using. That is kind of two dynamic programs, global and local alignments. And global alignment, we will see one algorithm as an example, Needleman-Wunsch, and local alignment, we'll see Smith-Waterman algorithm. And then matrices, some of the scoring matrices, we will see substitution matrices, amino acid and nucleotide matrices, example as PAM kind of thing and Blossom in amino acid. So these are the things we will try to see in today's lecture. So it has been estimated that over 12 million different species exist on our planet. The biodiversity across all the life forms, including plants, animals, and microbes, can be attributed to their unique genomic and proteomic composition. So it's understandable, right? Each one is having their characteristic, that characteristics based on their genes and proteins, what they are having. They're like an instrumental manual that guides about all the sequential tasks to be done in a tight order to accomplish uh, okay, Renupriya. Okay, noted. Um, like an instrumental manual that guides about all the sequential tasks to be done in the right order to accomplish a process. The biological organisms that have all the details in their genes, creating combinations of nucleotide, resulting in the diversity that we see in the biological world. So the difference in the sequences makes the diversity of the organisms, that gives the characteristics of the organisms, and that is what we can see the diversity in this biological world. So main thing is the sequence of the DNA and proteins. And the sequence of the bases in DNA has a crucial importance as it contains the code for the information diverse protein. Hence, the complexity on diversity of life. The unique order of bases in the results, DNA results in the creation of basic hereditary units makes genes. Sequence comparison of DNA can allow us to compare the differences at gene level across different organisms and species. So when you compare the DNA sequence, what happens? We can understand the gene level across the organism. So like, so if you want to compare with us with chimpanzee or another apes, so gene wise we can compare, we can understand how, what is the level of the difference, the genome wise. So first we have to start with the gene wise, and we can understand the different between the organisms and species. So comparative genomics is a branch of science that uses bioinformatics techniques extensively to trace the genes across multiple species and study their similarity and differences. So comparative genomics, especially another branch of bioinformatics, so that especially it will compare all the genes and genomes of the different species. So, in order to understand the differences and the similarities of the species, such studies help us in inferring the functional and structural characteristics of the newly found or existing proteins. So, when you have that kind of information, so when you find a new organism, let's say you found very newly new organisms which are similar to the humans. What happens when you already having existing information about the human and the chimpanzees or other apes? So when you find a new, suddenly you found a new organisms which is looks similar to the human. So what happens? You can align and see with the existing information, and you can find whether it is more near to the humans or more near to the apes. So that kind of just to give an example only, so that you can see more most of the times in the microbes. So there are so many microbes or insects which we are still not explored, so we are seeing. So for example, what we can see, what we have seen recently, the COVID-19, which is once it was sequenced, so it was found that it is more near to the coronavirus species which used to infect pangolin, and after that it's more near to the bats species. So that is how we can understand, once we have existing information compared to genomic analysis will give you information about the characteristics about the new genes or new proteins. So programmatically, biological sequence analysis is not much different from than the comparing strings and text, and thus developing concept of alignments is important. Sequences evolving over species and clades through mutations include insertions, deletions, and mismatches. So what are the units of this evolution, and how the different species evolving? So because of the mutations, that is mainly either insertions, that mean new nucleotide will be added, or deletion, that mean some nucleotide will be deleted. So both together we call as indels, or mismatches, that mean so uh, it will um, one nucleotide will be replaced with another nucleotide. So that is the mismatch. So this kind of mutations makes the evolving the new species. So that is what is still happening in case of virus. It's a very fast evolving, because millions of viruses can be produced within minutes. So um, so evolution also very more frequent than higher organisms. So thus you can see this kind of viruses evolving, that you can see in the news. So the COVID-19 is evolving into the news. It's not new species, but some of the mutations you can see. You cannot see it evolving fully. It is getting mutated. So when it is getting mutated in the proteins which are having the interaction with the uh, uh, more important interaction in the pathogenesis of the organisms, like spike protein. So that is more considerable, because it may change, that mutation may change to tightly bind than previous mutation, previous sequence, or that may leads to the loose binding. It may not able to bind uh, better than the previous one. So the worst thing is when it is changing to the not able to bind tightly than the normal one, the previous one. So it will not pass to the next generation, it will you will see the disappearance. And then the one will be having more, you know, spreading is the ones which are having the mutations which can tightly bind. So that is why we can see in the pandemics, again the peaks are increasing. Sometimes, you know, it may be bind more tightly than the previous one. So then it will you know, spread much more easily, and that is what happening. And so that kind of mutations are making evolving and into better species. So that is the, you know, evolutionary theory also, if you still remember, you know. So that is how it was believed that pangolin, when species coronavirus which are used to infect this pangolin uh, species, so it's accidentally those one, they used to eat in the provenance, some of the provenance of China. They used to have it, but they, if they have infected with the coronavirus species of the pangolin, and that used to be common a few since years. Even though it was infected, that would have been not, that might have not affected human, because that species was not able to interact with the human proteins, uh, is ACE2, for example. But sometimes it was, you know, one that 2019, what happened, it was got some mutations in that pangolin coronavirus species, so such a way that it could able to interact with the human ACE2 protein. And that's what happened, and that makes, that mutation makes whole world actually put into the, you know, very vulnerable situation. And yeah, very bad situation. So those kind of, you know, insertions and deletions and mismatches are the ones to evolve the species and clades. When comparing two biological sequences, an alignment is generated to view differences between the species at each position. So when you want to compare, so for example, pangolin coronavirus and a human coronavirus. So compare, what, how do you, how to compare? So you need to align. So you need to put that the pangolin ones and human ones one after one, and see each nucleotide and compare. And thus you will understand how much is the similarity is present, and how much are the the differences are present, and what kind of differences. It is the deletion mutations or insertion mutations or mismatches. So that is the pairwise alignment. And to just to give you an example, which is easily understandable in this particular situation. So come to the first homology. At the beginning, you know, homology mean morphologically, when we don't have much information about the genes and the genomes, and before the Darwin's era, what they used to have. So they used to have morphologically, okay, these two are homologically, they are having homology. For example, bats and butterflies, both will fly. Morphologically, they may thought both are belongs to the same family or kind of genus. Or at the time, no, you know, kind of genus kind of names, but they used to thought morphologically they are similar clan, but they are different, isn't it? Because bats belongs to the mammals and butterfly belongs to the insects. Bats fly and whales swim. So morphologically very different, right? Bats are flying and whales are swimming. Much morphologically different, but the bones in the bat's wing and the whale's flipper are strikingly alike. both belongs to the mammals. So bats and butterflies' wings are not homologs, and bat wings and whales flippers are homologs. So morphologically, at the beginning, before the much advancement of the science, before Darwin, that was the thing. Morphologically, we used to have the homology definition of the homology, and homology interpretation from Darwin to 21st century is Darwin explained homology as the result of descent with modification from a common ancestor. So even though there is not much information about the genetic material and DNA or RNA, but Darwin mentioned that homology is a result of descent with a modification from a common ancestor. So we are having the homology, that means we must both should have a common ancestor. So that is what he mentioned in 1859. And modern genetics say that homology information is in genes. Actually, the Darwin also said the same, but without knowing the information as gene. But modern genetics also says the same thing. It is coming from the genes. So two sequences are homologous if they are both similar and have common ancestor. Similarity itself is not enough. No, sometimes randomly, because millions of sequences of the DNA present as a genetic material. So random existing of the same sequence possible, if you take a short sequence. So similarity itself may not be enough. For example, if you take any of maybe five nucleotides, ATC, GC, you can say like a very short sequence. It randomly present in most of the organisms, in humans present and bacteria present, and sometimes any other organisms also present. So just similarity is not enough. So we should have a common ancestor. That is the main to compare the homology. Large enough similarities typically implies homology. So if you take larger ones, definitely it may be not randomly present that sequence in two different organisms or species. Mostly it is coming from the common ancestor. Sequence similarity comes with a significance measure. So why do homologies? A powerful tool to compare new discovered sequence with the known genes. So if you have found a new gene, so for example, again, you take the same, the coronavirus spike protein. So you compare with the new discovered sequence with the known genes. Okay, both functional and evolutionary information can be inferred. Regions of similarity is in unrelated proteins may be detected. Classical example. In 1983, after starting sequencing the information, the establishment of relationship between cancer and uncontrolled cell growth. For example, sequence of a new gene related to cancer was aligned against the sequence of genes known to affect the cell growth. So it was known information, some of the genes which are affecting the cell growth, and when it is aligned to a gene which is related to the cancer, the similarity between the sequence found to be very high, suggesting that cancer related to gene also affecting the cell growth. So thus they understand that by comparing those two information, that that comparison gave a new insight that so the genes which are affecting the cancer is affecting the cell growth. So that was another discovery and published in the Science in 1983. So this is an example, classical example. There are so many examples which gives you information, very important information by comparing the two sequences. So this gives you a, some kind of very commonly using the terminology in the homology. So come with the early globin gene. So you know, right? Hemoglobin is having globin, heme and globins, globin proteins, and globin gene is the responsible for globin proteins. And early, our ancestor, that is the globin gene, was only one, and somehow it is get duplicated due to the different reasons, due to some jumping genes, and yeah, due to some reasons, and it become, two different genes and having some differences, due to when it is getting evolved, some mutations it will have, and that mutations will be passed to the next generations. Thus the globin gene become two genes, that is alpha chain and beta chain, and this alpha chain was passed to the frogs and chicken and mouse and humans, all these things. And beta also passed to the mouse and chicken and frog, everything. Okay. And before this, frog evolved, so this the duplication was held, and this frog alpha, chicken alpha, and mouse alpha. So these are all will be the same locus, the same locus, and it will be having, having some mutations. But compared to all these three, will have some mutations, but all are, comparatively more similar than the beta. So these three are, I mean, whatever the organisms are having alpha, those we call as orthologs. So what is ortholog? Mouse alpha and chicken alpha are called as orthologs. And frog alpha and chicken alpha is called as orthologs. And chicken alpha and human alpha is orthologs. So whatever that is alpha between the different species, those are all orthologs. And mouse alpha, mouse beta. Within the mouse, it is having alpha and beta. Maybe alpha is in one chromosome, beta is another chromosome. These two are called as paralogs. So the same kind of gene which is present within the same species, they are called as paralogs. So not only the mouse alpha and mouse beta is paralogs, mouse alpha and chicken beta are also paralogs. Okay. So that is what the difference between the orthologs and paralogs. But all these collectively called as homologs. They are having some homology. So mouse beta, mouse alpha, when you compare, these are all homologs. Chicken alpha and mouse beta is also homologs. So, but all these collectively you can call as homologs, but there is some difference within the homologs. Those are, some of them are called as orthologs, some of them are all paralogs. I hope you understand the difference. So what makes the homology sequence different? So due to the mutations. So if you take this a short sequence to just to um, easy to visualize and easy to understand. So you have this gene ACGGC. It's not a gene, just sequence ACGGC, and it can get mutated into AGCGGC by insertion. One G is present here between inserted between A and C, and deletion, one G between G and C got deleted, so become ACGC. Or substitution, the first nucleotide A becomes C, so CCGGC. So these are all the kinds of mutations which makes the evolution. Insertions or deletion and deletions are the reverse of the another one, and the reverse referred as indels. Sequence alignment. So let's say this is the one sequence TCATG, is another sequence is CATTG. If you align these two sequence, what you will get? Maybe if you have a pen and paper, you try to align these two sequence and see. This is one sequence. You want to align with the another sequence, and how you can align. So T is not aligned with C. C is not going to align with A. A also cannot with T. T with T is okay. G with G is okay. Or you can have better. So by using exactly this one, you will get only two nucleotide similarity, right? But in fact, you can make it into better if you put like this. T is not aligning with anything. That means a deletion is here. That means is a gap hyphen. Usually here, nothing is there. You can put a hyphen here. And C with the C, A with A, T with the T. And this T is not having any pair. So it's a gap here. So that is a deletion here. And G with G. So when you put this, you are having four similarities and two deletions. One deletion here within T and G here, and one deletion here before C. There is nothing for the T. In another possibility is, of course, T with the nothing, C with the C, A with the A, but the T is aligning with this T because T you can align with this T or this T. It doesn't matter for us because both are having the same weightage similarity. But here the deletion we thought between this T and G in this alignment, and here if the T is aligning here, the deletion could be between A and T, and which is not going to align with this T. Both are possible. So both are having the same weightage because both are having four similarities, two possible deletions, right? So if you have multiple possible of alignments, how you can come up with the which is the best alignment? So that is the main thing here. We need the proper pairwise alignment methods. That's why the different algorithms was actually uh, developed to better uh, align the two sequences to find the similarities and differences. So how we can view pairwise alignment? Each pair in the alignment receives a value depending on the content. See here, in this possibility, you align as it is like this, not here, not like this. Just as it is you put C, T not aligning to C, that mean mismatch. C is A is mismatch. A and T mismatch. And T and T match. G and G match. So two matches are there. Three mismatches are there. Here, four matches are there. Two deletions are there. Here one deletion and one deletion. Here four similarities are there. Two deletions are there. So how you can say without without any thought, this and this are better than this comparison? How you can say? Because these are having more matches. Okay, so that is how we give a score. A score will be given for the identical character matches. You give positive score, and mismatches give some negative score. Not only the mismatches, the gaps, that mean insertions and deletions, we give also negative score. We give that kind of score, what happens? The same thing, okay? The same thing, T aligning with a gap, that means it's not aligning with any nucleotides. This T also with a gap, and the C to C, A to A, T to T, G to G. If we give positive score 1 for the matches, that means one match, second match, third match, and fourth match. That means four identicals are there, identical, sorry. So 4 plus 4. And indels here -1, and this is another -1, -2. So 4 minus 2 is the score is 2 here. And here also, here I think the colors was not properly put here. Okay, so here also 4 matches and 2 indels. So 4 plus 4 and -2, that is also 2. So these are all both are having the similar or identical weightage with the score wise. Okay, than this one. This is the what is the score. So T to T is 1, G to G is 1. So that is +2. And these are all mismatches, -1, -2, -3. So +2, -3, -1. So this is definitely having a low score, -1 compared to the +2. You understanding? So that is how the pairwise alignment can be done, giving a score to the alignment of the two sequences and what is the best alignment it will give. So this is not the best alignment. Either this or this is the best alignment, giving the best score. Two ways of for measuring homology between sequences. Given two alignment sequence, check how similar they are by counting their matches, or check how distant they are by counting their mismatches and the indels. How your perspective. So you want to see the uh, two given sequences, query sequences, how similar they are or how distant they are. So how similarity is equivalent to how low distance. So they are more less distance, they are more similarity. When the two sequences are more distance, that means less similarity. Similarity can be either positive or negative. So when you are giving a similarity score, just now we have given the score, right? So what we have given matches this much, mismatches this much, and indels are this much. You know, indels and mismatches usually negative score, and matches is positive score. And when you compile, I mean, summarize all those things, you may get negative score as positive score. Okay, just now we got a, one of the pay alignment you got negative score, one of the alignment you got, two of the alignments you got positive score. So similarity usually you will get either positive or negative score. Distance is always negative. So usually we will not do this, but just to give you, possibility, some analysis just to find how distant they are. So it will just give the negative scoring. Okay, identical scores have zero distance. So if you have fully identical, so let's say AT, GC, and you compare with the AT, GC. So you put both are 100% identical, so they have zero distance. So distance can be as low as high as 0. After that, it is a negative score always. Okay. Similarity between aligned sequences. The similarity score of an alignment between S and T is a sum of weights of each pair. Mismatches and gaps gives the negative weights, and matches always give positive weights. So the similarity between S and T is the score of the best alignments between S and T. S and T are the sequence names of, this is the S is ACTG here given, and T is given AGT. So previous slide again, the similarity score of an alignment between S and T is the sum of weights. So if you give negative weights to the gaps and mismatches and positive weights to the matches, and the best score will give the best alignment. So S to T, that means ACTG to AGT, you align. So this is the possible alignment. What is that? A to A, and C is not having any partner here, so put the gap, that is the deletion. And G is not also having nothing in the pair in the S sequence, that means put deletion. And T to come match with T, pair is there, and the G is not having any pair, put the deletion. So what is happening here? Two matches are there, three mismatches are there. Two matches minus three gaps. Okay. So these matches, you can give positive score, not always no plus one, and deletions not always minus one. So each kind of you know methods, pairwise alignment methods follow different values. Even the same algorithms, you can change your values to get to find the best match. Okay. So here, if you give the same kind of matches as plus one, mismatches are minus one, the score going to be minus one. Total score is going to be minus one because plus two, minus three. Sorry. Or you give matches as plus two, indels are minus one. So matches are plus two. Each one is plus two. So A to A is plus two, T to T is plus two. That means score is, I mean, the matches, the score is plus four. Mismatches score is minus one, minus one, minus one is minus three. So plus four minus three is going to be one. Total score is going to be one. So the match, the value as mismatch values not always plus one and minus one. So it you can it will change based on the algorithm we used and the alignment method we use. So distance between aligned sequences. So if you compare these two sequences in the distance score, what happens here? Uh, S is the ACTG, T is the AGT. The different mutations types have different weights. Distance score is equal to the minimal sum of mutations weights transforming S to T. So what is happening ACTG to AGT? So just to see only the S sequence and T sequence, what is happening? S is ACTG and T is AGT. What is expected when you are aligning these two sequences? So S becoming T sequence during the evolution. How best possible way it become, okay, and that means less steps. So how it is, ACTG become could be become AGTG, that means C become G, uh, that is the substitutional mutation. So instead of two indels, that mean you can C delete and G insertion, you can have the substitutional mutation. So C become G here. So one mutation here, and then AGTG need to be become AGT, right? So last G become deleted, so become AGT. So what happens? One substitutional mutation, one deletion mutation. So one substitutional mutation, you give negative value, one deletion mutation, you give another negative value. So overall, the distance score will be negative score. And the best case, if you compare fully identical sequence, let's say if you are comparing ACTG and ACTG, so what is a distance score? It's going to be zero. There is no distance score. I mean, there is no mutations. There is no negative value. So the distance score always either zero or less than zero. But the similarity score is either, sorry, similarity score is either negative or positive. Okay, this is one of the way how two sequences can be checked. This is called as dot plot. Okay, what is this? Sequence 1, all the nucleotides can be put in the Y column, each box. In another sequence 2, we can put in each box in the X axis. And what you are going to do, so just to compare the boxes, each box resulted between this and G. What is this box actually trying to compare? G and T. G and T are similar? No, don't put a dot. Then here, go to this box. A and T, not similar, don't put dot. A and T, no similarity, don't put dot. T and T, similar, put a dot. C and T, no dot, no dot, dot. AT, no, AT, no. CT, no. GT, no. TT, yes. So similarly, you put all the dots in all the resulted boxes, you will see this kind of view, and you can see some kind of line. It gives you the similarity. So how similar? If it is all the dots are filled, then it is fully similar. So otherwise, see, A and A is same, A to A, T to T, G and C not same, G and G, T and T, C and E not. So it will give you, this straight line will give you, and where it is having the similarity, and also give you how similar they are. This is the graphical representation. And this will give you so much of ambiguous, and it will give you so much of similarity within the same sequence. So you can change. There are some modifications can be done to find the better way. The same sequence, the sequence 1 and sequence 2, same sequence which you have seen in the previous slide, but now the criteria is different. The criteria is, when you are in this box, whether to put the dot here or not, the criteria is not to compare just G and T. Okay, you need to compare from this G. You take four bases or nucleotides, GAAT, and from here in y-axis sequence 1 also you take 4 nucleotides, TAAT, and put all these 4 exactly below to the 4. Don't align the best way, just to put uh, 4 nucleotides just below the 4, this sequence 2 4 you put GAAT, TAAT. Out of these 4, whether 3 are matching or not, out of 4 3 matching, put a dot, otherwise don't put a dot. Understand? GAAT, TAAT. So AAT are matching. Only G and T are not matching. So 3 out of 4 are matching, put a dot. Okay. And the next box, AATC, and here TAAT. Exactly put there AATC. Okay. Let me show you this. Okay. Okay. Oh, we have taken this one. Okay. Okay, first box, so you can see G and T, right? So what we have seen, G, A, A, T, that is the sequence 2, and the sequence 1 is T, A, A, T. So 3 out of 4 are matching, yes or no? These three, these three are matching. In case of this box, you take the second box, or maybe you can see the T here, where you have dot. Okay, so T, C, G, T. You take another one, T, C, G, T. Here, T, C, G, T. Then here, T, A, A, T. T, A, A, T. So the best alignment, you always use Courier New font. So all are same size, all alphabets. So easy to align the space. Here T to T is okay, C to A is no, G to A is no, T to T is no. So even though the T and T is matching, we put the dot here. But when you compare here, when you the new rule, that means 3 out of 4 need to be matched, but that is one, it is not fulfilling. Only 2 out of 4 is fulfilling, so we don't put the dot. Understand? So don't put here the dot. That is what happens in the next slide. So we didn't put the dot here. So we are going to put the dot wherever it fulfills the criteria 3 out of 4 bases are matching. So then you will get a better visual view that these two are more similar, more matching. Okay, and here a partial internal sequence also same. Okay, and this is the software it will give you, and how the two sequences are similar or not, whether it is where it is similar. Sometimes this one, this diagonal one, it may not be in the middle, maybe somewhere here. That means only this region of the sequence is matching with this region. Okay, if some if the diagonal, let's say if the diagonal is somewhere here, so what does it mean? This particular sequence is having similarity with this sequence, not fully match. Okay, but having here like this diagonal, what it is showing, this sequence is having similarity with this, but this is no line. That means this sequence is not having similarity with this. That is what it will show. Okay, so that is the dot plot comparison. So whether I gave or not, I will give you the practical 3 manual to you. So where it will be having a 1 to 4 exercises based on the dot plot you need to do. So then 25.